Thank you for the kind invitation. I would like to thank the organizer to have me here. And I'm very glad to meet the colleagues from ASM side. We just met 2 days ago at the Institute of Microbiology. And I still remember that Dr. Bertuzzi gave a very impressive presentation. Okay, thank you. My name is Linqi Wang. I'm from the Institute of Microbiology, Chinese Academy of Sciences. And my lab focuses on a clade of dangerous human pathogens. They are human pathogenic fungi, and which cause more than 3.5 million deaths every year, and with very high mortality rates, above 20% in average. In addition, the number of new fungal pathogens has risen sharply in the last 2 decades. So furthermore, and these slides can tell us another important information about human pathogenic fungi. That is, we can see, okay, the most fungal species in these slides are marked in red. So this means they are not obligate pathogen, are not commensal pathogen. Rather, they are of environmental origin, and they are called the environmental human fungal pathogen, and they are the major group of human fungal pathogens. Okay, so our lab focuses on a central question regarding environmental human fungal pathogen. That is why these environmental fungal species can cause fatal infections and be difficult to treat. Okay, as I just mentioned, the invasive infections caused by environmental human fungal pathogens usually have very high mortality rates. One reason is failure of antifungal treatment is very common in clinics. I would like to take cryptococcal meningitis as an example. And this is one of the most dangerous fungal diseases and mainly caused by the environmental human fungal pathogen Cryptococcal neoformans. And the Cryptococcal neoformans is the #1 pathogen on WHO human fungal pathogen list. And amphotericin B is the only available fungicidal drug to treat Cryptococcus infections. And the other drugs, they are the statistic. That means they can only inhibit the growth of Cryptococcus cells and cannot kill them. Okay, so if amphotericin-based therapy fails, so the mortality rates can be as high as 70%. It's very dangerous. Okay. So in 2006, and Dr. King Lewis lab published a paper in ASM journal, AAC. Okay, this is an excellent journal, right? So I guess the doctor, the Iris, would completely agree with my view, right? Okay. So in that paper, and they raised a hypothesis. They think a specific form of drug resistance called phenotypic resistance is an important contributor to antifungal treatment failure. So what is a phenotypic resistance? The phenotypic resistance is not related to a resistant mutation, but describes the phenomenon in which genetically susceptible pathogen cells surviving high doses of cyto drugs without a change in MIC. But the field of fungal phenotypic resistance has not been really established in the field of fungi because there is no evidence supporting that the fungal phenotypic resistance can be induced by host factors during fungal infections. And if you can identify such evidence, it's very important for the establishment of this field. Okay, so what is the bottleneck in this field? Okay, the bottleneck is how we discover host factor that can strongly stimulate fungal phenotypic resistance. Since we have a lot of members of host factors in our body, right? So to this end, we develop a systematic approach which allows us to perform a comprehensive evaluation of triple interaction between metabolite, drug, amphotericin B, and Cryptococcus cells. And based on this method, and we identify some host metabolites can strongly stimulate phenotypic resistance. Among these the metabolites, we found glucose has the strongest effect. Okay, we were very excited about this finding. Why? Because so our brain is rich in glucose, right? Because glucose provides energy for our brain. And Cryptococcus neoformans happens to be the leading pathogen of fungal meningitis. Over 70% of fungal meningitis cases were caused by this pathogen. So next, we want to know whether or not the presence of glucose in our brain is sufficient to trigger phenotypic resistance in Cryptococcus neoformans. So we collected CSF samples from different patients, including ones with Cryptococcus infections or other diseases. We also use an enzyme. This enzyme is named glucose oxidase, GOX. GOX can catalyze the conversion of glucose into gluconic acid. And based on our systematic approach, unlike glucose, the gluconic acid cannot trigger phenotypic resistance in Cryptococcus neoformans. So this enzyme-based approach basically allows us to directly test whether or not the presence of glucose in CSF samples is sufficient to trigger phenotypic resistance. Okay. So we found when you remove glucose from CSF, okay, the level of phenotypic resistance decreases dramatically. And if you add sugar back, the phenotype comes back. So, and this data strongly suggests that glucose is the key factor in the driving phenotypic resistance in human CSF. So next, we want to know what is the main factor in mediating glucose-induced phenotypic resistance. Okay, as a result, we identified the transcription factor MIG1. Actually, it's very easy to figure it out because MIG1 is very conserved in fungi and plays a conserved role in mediating phenotypic, in sensing glucose. So if you knock out MIG1 in Cryptococcus neoformans, so basically the glucose-induced phenotypic resistance is blocked. Okay. So next we will ask the most important question in this project. That is whether or not MIG1 mediated phenotypic resistance can eventually compromise the efficacy of amphotericin B therapy against Cryptococcus meningitis. And if answer is not, we don't have to bother with such phenotypic resistance, right? Because it may not have a clinical significance. Okay, so to this end, we use the mouse model of Cryptococcus meningitis and infected with a virus type and the MIG1 mutant strain. And we found that without amphotericin B treatment, and basically there is no difference, significant difference in terms of mice survival. Okay, so this suggests the MIG1 is not important in brain infections by Cryptococcus neoformans. But if you treat a mice with amphotericin B for 2 weeks, then you stop the treatment, you can see the big difference. Okay, so in mice infected with viral type cells, and after stopping treatment, okay, all mice died within 3 weeks because of relapse. Okay, but in mice infected by with MIG1 mutant cells, the 60% of mice were cured by 2 weeks treatment with amphotericin B. Okay, so that means we cannot detect any living cells in their brain. Okay, so this data demonstrates MIG1 mediated phenotypic resistance affects the therapeutic efficacy of amphotericin B against Cryptococcus meningitis. Next, we want to know the mechanism underlying such a phenotypic resistance. Okay, so as we know, so amphotericin B kills fungal cells through binding to membrane component ergosterol, which is the target of amphotericin B. Okay, we found in the presence of glucose, the transcription factor MIG1 can promote the production of another membrane component named IPC. And the IPC uses a type of a war mechanism. And this component can directly interact with ergosterol, the targets of amphotericin B, and competes with amphotericin B for ergosterol, which make a phenotypic resistance happen. Okay, so since we already figured out the IPC production is essential for the glucose-induced phenotypic resistance, okay, so we hypothesize the inhibition of its production may enhance the efficacy of amphotericin B against the Cryptococcus meningitis, okay? And we used a inhibitor of the fungal IPC synthesis. And this, the compound name is named Aureol Basidium A, ABA, okay? So ABA has a very nice safety, but it was eventually abandoned by Merck company because of its narrow, very narrow fungicidal spectrum, okay? But we found the amphotericin B and ABA combination shows a promising efficacy against the Cryptococcus meningitis. So, and if you still can remember, and I mentioned earlier, the Cryptococcus meningitis is one of the most dangerous fungal disease. And 7 days of treatment cannot result in a cure with any clinical recommended strategies. But when you treat mice with amphotericin B, ABA combination, and the cure rate can be as high as 70%. So this number is very promising, okay? So in addition to brain, our lungs are another organ commonly infected by human fungal pathogens. But unlike brain, so our lungs are glucose poor, okay? And our amphotericin B killing assay show that the phenotypic resistance cannot be induced by glucose at lung-like concentrations, okay? But it has been well established fungal cell in lung infections are also difficult to clear by amphotericin B. And this suggests phenotypic resistance may also occur during fungal lung infection, but the mechanism is totally different from the one we identify in brain, okay? And my postdoc, Wei Xin, noticed a paper from Dr. Drummer, and they raised a hypothesis in the paper. They believe Cryptococcus neoformans can form dormant cell population and during lung infection. So we all know that in bacterial, the cellular dormancy can also promote phenotypic resistance. And the dormancy mediated phenotypic resistance is called drug persistence. Okay, so we ask question, whether Cryptococcus neoformans can form the amphotericin B persisters in vitro or during lung infections. We test this idea according to the definitions of drug persistence. First, the cells don't undergo proliferation because they are dormant, right? And if for Cryptococcus neoformans, it's very easy to collect dormant cells. You only need to grow Cryptococcus cells to stationary phase. Then you can look at, you can get the dormant cells look like this, look like a bees without body. For the growing cells, you can see budding there. This means they are proliferating. Importantly, the survival rate of dormant cells is 100 to 1,000 times higher than that of growing cells. There is less than 1% of cell population surviving, and there is no difference in MIC between growing cells and the dormant cells. So our findings consist with the definition of drug persistence, and this data suggests Cryptococcus neoformans can form amphotericin B tolerant persisters in vitro. Next, we ask whether such drug persistence, amphotericin B persistence, can occur during Cryptococcus lung infection. Okay? So to do this, and we take advantage of multi-omics approach and identify a protein. We name this protein SPS1. SPS1 is dormancy specific, and this protein represents the most abundant protein in dormant Cryptococcus cells. And so the gene encoding SPS1 can be used as a reporter gene. And we infected mice via inhalation with Cryptococcus cells carrying SPS1 based reporter system, okay? 7 days after infection, we can clearly see a population of fungal cells highly expressing the dormancy specific protein SPS1 in the lungs. And these cells were sorted by flow cytometry and subjected to amphotericin B killing assays. And we found SPS1 expressing dominant cells are highly tolerant to amphotericin B. So this data suggests amphotericin B persistence also occur during Cryptococcus lung infection. Okay, since we identified a way to evaluate the formation of amphotericin B persisters in vivo and in vitro, And this allows us to explore the drug against amphotericin B persisters. Okay. So, and in this regard, we use a drug repurposing approach and identify the antidepressant, the sertraline, and which has a very strong effect on killing amphotericin B persisters in vivo. By the way, and Dr. Bingjai is the first one identify the anti-cryptococcus activity, and she's sitting there. Okay. So the last part of my talk is regarding another issue related to environmental human fungal pathogen. So that is in the last 2 decades, we can see many new pathogenic fungi have emerged. And importantly, all of these new pathogens are of environmental origin. So the question is, what role do environmental factors play in the emergence of pathogenic traits in environmental fungi? Okay, this is a very important question. In collaboration with the China Hospital Invasive Fungal Disease Surveillance Network, and we identify a novel fungal pathogen. Okay, so I'm sorry, already I have published the paper, but I still cannot make correct pronunciation of this species. Okay, so I just use RF to refer to this novel pathogen. Okay, so and this pathogen is multi-drug resistance pathogen. Unluckily, 2 patients infected with this pathogen died because of treatment failure. Okay, so we use the drug repurposing approach again and identify the bacterial antibiotic Polymax B, so which is very effective in killing this new pathogen. And interestingly, we found that rising temperatures can strongly promote the mutagenesis and enabling the emergence of hypervirulent and pan-resistant virulence. So this pathogen is very dangerous. Okay. And we used the animal model and found the variants with hypervirulence can even cause death of immunocompetent mice. Okay, so this data support the possibility that the global warming may favor the evolution of this new pathogen. And this data also, okay, so also trigger public interest and discussion about global warming driving fungal evolution and emerging fungal diseases. And our lab is currently testing approximately 700 fungal species. And for rising temperature, the effect of rising temperature on mutagenesis, the one by one, okay? So hopefully I will have some data to support this idea, okay? So I would like to thank my collaborator and the lab members, and also I thanks to my daughter. I paid just under $1, and she drew the portraits of all the lab members, okay? So I think this reflects the theme of the symposium, okay? That is, we need collaboration between scientists from different countries and to collectively advance the field of microbiology. I just borrow the words from Bertuzzi, Dr. Bertuzzi, 2 days ago, okay? So thank you very much, and I would like to take questions.
Thank you for that great talk. Questions? Maybe I'll ask one while we're getting going. The rising temperatures increasing mutagenesis, is that due to increased replication of the fungi?
I'm sorry, can you repeat that?
The increased mutagenesis that you get with rising temperatures, is that simply due to increased growth of the fungi?
I think the growth affect the mutagenesis because, so for fungi, it have to replicate and the mutagenesis can happen, right? If it cannot grow, so the mutagenesis cannot happen, right?
Okay, and some of these drugs that you're repurposing, did you see broad spectrum activity or is there a lot of specificity to the different fungi?
Oh, you mean the Polymax B?
Yes.
Oh, Polymax B actually, Polymax B also being found antifungal, is antifungal activity. Okay, so the Polymax B is very useful for this new fungi, but for some common clinical species like Candida albicans, I think it's not has a very good effect on that. Okay, very good question.
Other questions? Great.
Okay.
Great talk, Linqi. Have you found any evidence of plant fungal pathogen becoming human pathogen?
It's very very difficult to achieve that because it involves so many factors like ecology, biology, and even human activity. Okay, I think we may need a lot of evidence to answer this question, but just based on our finding, I think it's not sufficient to support. So the temperature, actually rising temperature, makes this environmental fungus become a new pathogen.
Okay, thank you.
Okay.
I got one. So thank you very much for this beautiful talk. So in clinical practice, we know that when you have cryptococcal meningitis, mostly immunocompromised patients that do not have cellular immunity, and you take the CSF of those patients, you usually need to do combination therapy, which we normally do with 5-fluorocytosine plus amphotericin B. But the glucose of these patients are usually very low in the CSF, right? And I'm just trying to reconcile that with your data of the mutant and this glucose effect.
Okay.
Can you just expand a little bit?
Oh, okay. So this is a really good question. This also the question, is ad hoc reviewer asks us. Okay, so this is the reason why we involved CSF samples, the CSF samples from patients with cryptococcus infections, actually. So we found even for the CSF with a severe cryptococcus infection, the glucose concentration is sufficient to trigger the phenotypic resistance. Okay, thank you very much.
In the case of lung infection, you propose that dormant fungi could lead to the infection. But how do a dormant pathogen, cause infection? They didn't divide.
Oh, this is a good question, though. I think, the Dr. Jorma's lab think the dominant cells serve as a reservoir for latent infections, actually. But, so when our immuno condition changed, actually, maybe the dominant cells can be reactive. So, and the reviving the cryptococcus cells can cause the acute infections. That is fatal.
So have you checked this dynamics in your mouse model?
I checked. I did check. And we sought the dominant cells and from lungs. And we infect mice with dominant cells. And those cells, just reactivated and caused the fatal infections in mice. So I didn't show the data. Okay, good question.
Thank you.
Okay, thank you for that great talk.
Thank you.